Thank you for the nice introduction. I'm the head of the Bioinformatics program actually at School of Medicine at Acıbadem University. And I'm also the founder of 3 companies. Epigenetics is one where we give the support to clinicians for personalized medicine applications. And I will give some brief introduction about the Omix technologies. With the advancements in the omics technologies, starting with the NGS technologies, which was initiated by the Human Genome Project, now we have access to different types of data, and that's changing the medicine drastically today, because in the past we were basing our decisions based on the things that we could measure, and those things were the symptoms. And you have high fever, you have a rash, you have this, you have that, and your blood sugar level is this much. And those were the things that could be measured, and the clinicians were making their decision based on these because they didn't have any cellular level information. But thanks to Human Genome Projects and the other projects like transcriptomics, proteomics, glycomics, metabolomics, and so on, now each of these data is giving a glimpse at different things that are going on at the cellular level, and that can help us to understand the disease development mechanism. For example, genome sequencing gives us the genotype information, and then from then we can identify certain mutations, and that can give some prevalence for different types of diseases. And there are several databases like ClinVar that are updated regularly, and you can find associations between the variants and the diseases, and that can be used for diagnostics as well. And we have transcriptomics databases available, and we can measure the transcript levels, and using RNA-seq technologies especially, which can also determine which splice variant, which transcript generated by that gene that might be responsible, which was not possible with the old technologies like microarray technologies, because there you could determine the mRNA level, but you couldn't determine the transcript level changes. And sometimes mRNA doesn't change, but a certain transcript is being expressed, and that's responsible for the disease. So this technology today, using the sequencing technology, RNA-seq, enables us to determine such differences and helps us identify how these things can play a role in disease development mechanism. Today, we can measure the protein data highly accurately, and plus we can determine its functional status. So maybe you don't see any difference in the blood serum, for example, of a certain protein, but its functional status is different. One is in the disease individuals, it's phosphorylated. In the healthy individuals, it's not phosphorylated. So not the level, but the functional status can be causing the disease. So today's technology enables us to obtain such information as well. We have ATAC-seq sequences where transpose accessible chromatin regions are exposed, because we know today that the chromatin folding process, epigenetic mechanisms play a key role on the transcription process, which genes will be transcribed, which genes will be produced, and so on. And these chromatin modifications plays a key role in this case, and as well as in the development process. So that's why you have to take these type of information into account as well. What happens to the chromatin regions, open chromatin regions, close chromatin, how these changes with the disease. So ATAC-seq data enables us to follow this. ChIP-seq data, it's chromatin immunoprecipitation followed by sequencing. So you can determine the DNA binding regions, the certain transcription factor binding sites and regions, and which parts are transcribed and which transcription factors are binding to which DNA sequences. And you can determine through this the chromatin level changes as well. We have single cell RNA sequencing technologies. Recently, we see that it's crucial to identify what's happening at single cell level, to understand the processes. Especially in cancer, there are several applications. And we have several ongoing EU projects on this one to determine pathogenicity, how pathogens become pathogen, or cancer stem cell. How does the cancer stem cell turn to cancer? And we have lots of single cell sequencing and also epigenetic data gathered to understand this transition from a single cell to pathogenic cell, like for certain known pathogens and viruses and so on, how this pathogenism mechanism is developed. And for that, you need the single cell information. And so all this type of information giving us different level of information about the process. We have also the methylation, which is very important, especially for many diseases. It's a dynamic process, and especially the regions, upstream regions of the genes have the promoter sequences, and those regions have abundances of CG letters. That's why they are called CPG islands. CG repeats, and the C is methylated. And when it's methylated, it's believed that since this is the promoter region is covered and transcription factor will not have access to that site, and that will influence the regulation and transcription of that gene. And there are some experimental evidence against it for some exceptions, but the general belief is that if the CPG islands are methylated, the gene that's coming afterwards is not going to be transcribed. And if it is unmethylated, then it will be transcribed. But the key thing is this is a dynamic process. So it can change with your stress level, it can change with your diet. Many things can influence these things. So for example, a serious event happened, you lost someone suddenly and it caused a sudden stress, and these kind of events can change the methylation status drastically. And the genes that were turned off are now being turned on and vice versa. So this is a major event and it causes a major consequences in the health. So this may play a key role in many diseases, and you have to take this into account as well to understand the disease development mechanism for different types of diseases. So you have to integrate all of these. Plus, now we know the importance of microbiome because we don't live alone. We are like incubators for several microorganisms in our body. Actually, we are more bug than human cells. This ratio is not correct anymore, but still we know that there are so many different organisms, so many different cells. They live in harmony with us and we communicate with them. They support us in many ways and we provide them the necessary environment, the food and the temperature and everything they need to survive. So there are 10 to the 14 microbes living and up to 35,000 species. Majority of them we still don't know, and they contribute a lot to our health, and they carried out some experimental status. They took the gaita of the obese twin and replaced it to the lean mice, and the lean mice started to get fat. And they did the opposite experiment. They took the gaita of the lean twin and transplanted in the obese mice, and the mice became lean. So that means there's this crucial information there, and this system, ecosystem, is communicating with the body and creating a big response as a result. So we have to take into account microbiome in many diseases and many drug response mechanisms. And right now we have two ongoing projects. One is to understand the drug resistance in epilepsy and the role of microbiome in drug resistance. And another one is the cancer therapy in colon cancer and the role of microbiome in the drug resistance, therapy resistance. And there are key factors involved. So you have to take this into account as well. So there are so many levels of omics information that will give you glimpse of pictures of different parts of the body, how it responds, how they communicate. But the key is how to integrate this data to understand what's happening in that person's body. And for that, we need AI models, and the models that we can use, like network theory, network models, and so on, to incorporate this data. So that's the big field right now, AI in translational medicine. There was a recent ERA-PERMET call on this one, building AI models and translate it into medicine. And AI is basically learning from the available data. So from the big data, you can learn certain patterns and use those patterns for diagnostics, and as you can use them as biomarkers, you can use them as factors that contribute to the disease development mechanism. So they help you to understand the individualized disease mechanisms as well. So, and as a result, you can combine complex data levels. You can combine the clinical information, genetic information, and other in all the omics data using AI models, and build models that were discussed early on in the morning, like if this gene is overexpressed, and if this person has high heartbeat, irregular heartbeat, and if the blood sugar level is blah, blah, then this disease is the most likely diagnostics for this person type of thing. So you can bring in together different level of information using the AI models. We do network-based integration of omics data. We developed a method where we map the variants of an individual, all the omics data. We started with the SNP data, but we translated this into all different omics data. We map all the variants and their impact, because not all the variants are impactful. They are not disease causing. So we also give an impact score for every variant, even at SNP level or transcript level and methylation level changes. Not all the changes are disease-causing changes. They contribute minor amount to that individual's health status. So we identify the key ones and give the impact score. And based on that, we map them to the genes that they are affecting. So we take the human protein-protein interaction networks. In these networks, they have the interaction data of the proteins, and these, here you have to use you have to be extra careful, use the reliable networks, because we have so many unreliable interactions, unvalidated interactions in certain PPI networks. So you have to build your own network for your disease, where you know that these proteins are actually interacting each other to carry out certain functions within the cell. So we take those protein-protein interaction networks and we map the affected genes. So here the red ones are the disease-targeted genes in this individual, and the blue ones are the ones that they're interacting with, but they are not affected in this individual. So, and the size of the circle gives us how badly it's affected in this individual. So then we find active subnetworks. We have our own algorithms to find the active subnetworks, which is most of the red dot containing subnetworks. So that means these interactions are heavily affected in this individual, either through methylation change or a SNP or a chromatin binding site, or changing the microRNA binding site, and so on. It can have several levels of changes. So, and then you find a subnetwork containing most of the red dots, and then find out which pathways, which signaling cascades are affected as a result of these changes in this individual, and that helps us to understand the disease mechanism. We applied this to epilepsy data initially to show that this works, because I don't believe in, especially for complex diseases, the disease development genes are not identical. It's a complex disease. It wouldn't be a complex disease otherwise. So different things are affected, but the ultimate change should be at the cellular level. The end result at the cellular level should be the same. So same signaling cascades, same pathways should be affected for that individual to develop that disease. So that's why we search for marker pathways. And I think that's the correct concept to study, to individualize these diseases as well. So we identified the affected pathways in epilepsy using this Omics data. And these, we assign significance values to affected pathways depending on how many genes in that pathway is affected. And most of these pathways were identified in the literature by experimentalists and clinicians as a disease-causing, epilepsy-causing pathways. So, and then we wanted to show that this works in different populations as well. So we got the data set from Yale group, where they conducted intracranial aneurysm analysis in European and Japanese populations. And when we carried out this study, we found out that there were only 15 genes that were commonly affected. There were only 6 SNPs that were common in both of them. But when we look at the affected pathways, 7 of the top 10 pathways were identical. So this cannot be coincidental. There are so many pathways, but when you look at the impact of the affected pathways in the Japanese and the European population, 7 out of 10, and the probability of this happening randomly is 10 to the minus 34. So it's not a random event. Same pathways, same signaling cascades affected in both populations so that they can develop aneurysm. And when we look at the MAPK signaling pathway, this proves our point as well, that the yellow ones are the genes that are targeted in the Japanese population, and blue ones are the genes that are targeted in the European population, and the red ones are commonly targeted genes. Only 2 commonly targeted genes, but as you see, several genes are affected in MAPK signaling pathway, resulting the malfunction of the MAPK signaling. And MAPK signaling is a key pathway for aneurysm. So it doesn't need to go through the same gene. By affecting different set of genes, if you alter the function of this pathway, then, and this is only one pathway contributing to aneurysm, there are other pathways, key pathways as well. If all of them are malfunctioning, then as a result, you develop the disease. So this enables us to individualize the individualized mechanisms of the disease development and assign risk scores. So we developed a scoring system for each pathway, how well it's functioning. And I know that I have a mutation in RAS-GRP. I know my genome sequence, and I have a mutation in TAB1, but these are not very impactful mutations. They don't actually do any harm to the function of these genes. So my MAPK signaling is intact. So it works more than 99.5% efficiency. So since MAPK is a key pathway, and I combine my signaling efficiency for other pathways for aneurysm as well, and from the combination of those, I assign a score for myself, for any individual that's carrying out such a study. And my aneurysm development risk is very low because most of the pathways that are playing a key role in aneurysm are functioning well. So you can assign such scores, and that can help you to individualize the pathways. And I can also find out which path is causing my MAPK signaling not to work efficiently as well. And that can give you the disease target, new disease therapy targets as well. And to this end, we conducted another study. Behçet's disease is a neuroimmune disease that's commonly seen in Turkish and Japanese population. So we got their data, and then we analyzed them. This time we didn't analyze only the pathway level, but within the pathways, there are several signaling cascades. We wanted to find out which signaling cascades were commonly affected in both populations to identify novel therapy targets. And we found out that 25 signaling cascades out of 40, top 40 were identical. Probability of this happening is minus 134. E to the minus 134. So this is not random. The same signaling cascades are affected for these populations to develop Behçet's disease. And JAK-STAT is a key pathway for the Turkish population. And in the Turkish population, cytokines, receptor, JAK and STAT was affected, GFAP was affected, and that causes differentiations and that causes a change in the cell cycle. And in the Japanese population, again, cytokines, it's not the same cytokines, different cytokines, but cytokines are affected, receptors are affected, JAK-STAT is affected, AUX is affected. Instead of GFAP, this time AUX is affected and it changes the lipid metabolism, but the end result is the effect on the cell cycle. So this similar part is the JAK-STAT pathway. This part is overactivated. And there are drugs targeting the JAK-STAT developed for rheumatoid arthritis. But those drugs were never used in Behçet's patients. And we have an ongoing project with them right now that they use a different therapy for Behçet people who are going through some attack periods. But there are people that are not responding to that therapy. We propose that the JAK-STAT blocking therapy, like rheumatoid arthritis drugs, may be a potential application for those individuals as well who are resistant to known therapy. And now they are conducting a clinical study and they are showing that it's showing a promising result. So if you know that this individual has its attack because of the overactivation of JAK-STAT pathway, you can block this one, but maybe through a drug that's designed for another disease. So that opens a whole new area of research of drug repurposing. So from now on, you're not going to have the drug for the disease, but you're going to have the drug for its target. And if that target is activated for any other disease like cancer, we know that there are some angiotensin drugs that are used in treatment of cancer cases. And there are some interesting publications on that one. If you can find a driver mechanism of an individual, maybe a drug targeting that individual is not available for cancer as a cancer drug, but another disease drug can create the same influence that you want in that individual, and you can use that as a therapy for that individual. We also applied our pipeline for several rare disease applications. And in this case, we prioritize, using our pathway-based methods, we prioritize variants of the individual based on the symptoms that they carry out and whether those symptoms are causing certain diseases as a result as well. So we have a variant prioritization algorithm taking into account this network structure. And we applied it to this case. This was a very challenging case, 6-year-old girl. And these are the symptoms the girl had, and she went through everywhere. She went to Mayo Clinic, she went to Germany to be diagnosed, and she wasn't diagnosed until she came us. And she had several variants, and the heterozygous variants and homozygous variants, we ranked them with pathogenicity tools and MGI and literature search, and we filtered it down to 6 heterozygous variants and 4 homozygous variants. And then we applied our algorithm to prioritize these, and we identified this variant. This variant was called VUS, variant of unknown significance, because it's a novel variant and its impact was not identified before. So all those people, since it was classified as a VUS, they didn't take this as a disease-causing variant, but most of our prioritization algorithm gave this as the key that explains majority of the symptoms of this individual, this kid, and it causes GLUT1 deficiency syndrome. And the way we found this is this kid also had hemangioma, and this gene is also involved in hemangioma development. So it could explain 2 of the features at the same time, and that's why it was prioritized high in our ranking. So then we validated with the mother and the father that through Sanger sequencing that the variant is present in the kid and not in the parents. So when we looked at this syndrome, all the symptoms match very well, but to validate it, we had to do the glucose test from CSF, and it was validated and clinically diagnosed as GLUT1 deficiency syndrome. And our publication is almost being published, so I can send you the links to this publication. And they applied ketogenic diet to the kid. That's the treatment for GLUT1 deficiency. And now the kid is seizure-free, decreased involuntary movements, more understandable speech, increased vocabulary. So this is a case where the network-based prioritization, symptom-based prioritization may play a key role in diagnosing rare diseases as well. We have several cancer applications. Oops, this slide is not very good. I love this paper by Stephens. It's a breast cancer patients. Please look at the figure from the paper. Somehow it didn't look fine here. Every column in this is a different breast cancer patient. Red ones are the somatic mutations. Blue dots are the copy number variations. As you see, the dots are everywhere. So this is a complex disease, of course. So different mechanisms are affected, but end result is breast cancer for all of them. So, and unfortunately, there are certain protocols. You give the most likely protocol. If the patient doesn't respond, you give the second most likely protocol. But you don't look at the driver mechanisms for that individual at the individual level. And majority of them get treated with the first protocol. They don't respond. Some don't respond, and they are given the second protocol and so on. But we score the driver pathways. We give a driverless score for the changes in that individual by incorporating the Omics data. We have a scoring algorithm that we develop, and we identify the key pathways. So these are the red stars are the genes that are altered in this patient. This is a real patient data, but the most affected pathway was the VEGF pathway. And for this individual, we offered a VEGF blocker as a therapy for this individual. In this case, the same pathway was affected, but the impact scores were much less. And the mTOR pathway was the most affected pathway for this individual. So we offered the mTOR blocker therapy, which was not the usual therapy for this patient. And in this case, the growth factor receptors were over-amplified. So we suggested a therapy targeting this site. So if you can identify which pathways are the driver pathways, you can assign treatments targeting the driverness of that individual. And this is a case of preventive medicine, why screening of individuals' genotyping is crucial. And in the future, hopefully, it will be more accessible and more cost efficient for everyone to do this. This kid had thymoma cancer. And when we did the tumor and the blood analysis, genotyping analysis, we found out that the kid has AMPD1 stop gain mutation and early on. So this gene was not producing any protein. And this deficiency is causing myasthenia gravis. So this is connected to myasthenia gravis, and most of the myasthenia gravis has a high risk of developing thymoma. So if this kid was genotyped early on and identified this mutation, and he was screened for myasthenia gravis all through his life, he could be checked for thymoma, and they could catch this thymoma early on and remove it and save the patient. Unfortunately, when he came to us, it was spread all over. So that's why I'm giving this as a preventive medicine application, that people should genotype, screen themselves, so they can be under constant control, and you can diagnose the disease, catch the disease early on, and you can prevent all these things to happen early on. So I think this is a great future application as well in clinical bioinformatics. So I think genotyping and prevention will play a key role in clinical bioinformatics. as I said, we are also studying the drug resistance mechanisms using different types of omics data, and especially the role of microbiome in the drug resistance mechanisms. I wanted to mention that. And these are the projects that we are involved in. And my graduate students, they developed all the tools. I thank them, and I thank you for listening.
Thank you so much for the talk. That was really interesting. Now I need a little bit support by Jolt, how I get to the questions.
Yep. What exactly is the question?
So how do I see whether there are questions, or how do I?
You have to open the Q&A, but there are currently no open questions.
Oh, no open questions. Okay, wonderful. So yeah, I do have a question. Is genetic testing routinely integrated into the clinic at your department, or is it still kind of on a case-by-case basis? Can you maybe comment on that a little bit?
It's not integrated fully, but for most of the rare diseases, childhood diseases, yes. If it's not clean, they do screening for certain types of mutations, but if they are not known, then the genetic testing is done. And now people are moving more to whole genome than the exome, because there are so many unsolved cases that cannot be explained with the exome sequencing only. So more and more the clinicians, geneticists, or the neurologists are moving into whole genome sequencing. And then they need us because they come up with so many variants and they don't know which ones to prioritize, how to use those variants and tie them with the symptoms and the diseases. So there, I think that's a whole new area of clinical bioinformatics, how to prioritize these variants.
Right. I do have one other question in terms of, you mentioned the mapping to pathways, right? And to kind of just figure out how these multi-gene disorders work. Does AI help in any way in mapping, essentially, you know, the multitude of SNPs, right, to some phenotypes? Does AI and interpretability or some other kind of technologies help you find new kind of combinations of SNPs or mappings of SNPs that would help you kind of dig a little bit deeper at how this all plays together, right? I mean, it's not necessarily having a knowledge-based approach like from the pathways, but more kind of a data-driven one where AI kind of helps you understand what's going on. I mean, is there any movement in that direction? Or
yes, I think so. I think as we get more reliable data, actually for AI to be successful, we need the clinicians and expert clinicians, expert annotators. We need reliable data because, as you know, garbage in, garbage out in the AI model. So it has to be very well annotated. You have to be sure that you have the correct phenotype information. Everything is very detailed, explained. If we can get such cases where we conducted one study that we have an AI model for drug resistance to colon cancer, and any bacteria doesn't show strong association, but combinations of bacteria shows a very strong association with the resistance mechanism. So if you look at single biomarker, you don't find it, but those associations, that intriguing intertwined interactions between the microorganisms and how they play a role in drug resistance, those things are picked up by the AI models. Right. So that's a data-driven thing.
Great. So I see two questions that I'd like to squeeze in, maybe three. Daniel, you want to start?
Sure. Thank you, Michael. My question would have fit perfectly between your two questions. So you just said about using AI to predict something we might not know from the raw data, but not maybe from what we've seen. So you said that you're using these these interpretations, this expert knowledge to diagnose single cases, how do you capture this information? And do you have a system in place that helps you in a follow-up in another case, that you can look up what you previously did?
Yes. So how we capture is, for example, again, this AI microbiome and drug resistance association study that we conducted, that we built the AI model, and the most successful one was the random forest model. And random forest, as you know, is filled with trees, and each tree uses their own set of combinations of features to decide on the drug resistance. Then we digged more into each tree and found out commonly seen features together, microbes together in the successful trees, the ones that are explaining the data more. And that enabled us to find out associations between different microorganisms. If they are frequently seen together in a good classifier tree, they are contributing together. So they have complementing information about the resistance mechanism. So you have the AI model, and then you do further analysis of your AI model to retrieve the information that you can use in the clinic, actually, because it has that information. You build so many trees, thousands of trees, and they all have very valuable information, and the clinician need information that they can relate to and they can modify. And then we say that, okay, Fusobacterium nucleatum is increased a lot in these individuals along with Alistepis. So, and they have such an association together, and that's why if you change the levels of Alistepes and Fusobacterium before the treatment, the patient would respond to the therapy more successfully. So the AI models, of course, you give in the data, you build your models, and there is so much information because it's like a black box for them. So you dig in more as a clinical bioinformatician to find out information that can be used in the clinic for therapy and diagnostics. And for that, you develop your own algorithms as well. So we developed this algorithm to find out commonly seen microorganisms in our random forest trees and use those as a feature. Afterwards, we built another model for that one. And we found out that actually we can actually find out how they interact with each other, these microorganisms, in the way to cause the drug resistance to certain chemotherapeutical agents. So that's how we do it.
Okay, thank you so much.